Let's go with Ria. Ria, you there?
Yeah, I'm here.
Okay, great. Go ahead. Do you want to just fix your... Yeah, that's perfect. Yes, we can see your screen. Yes.
Good afternoon, everyone. My name is Ria Sri Ramanathan, and I'm a freshman at Gillern High School in Albany, New York. My topic is blood-based biomarkers in Alzheimer's disease, a systematic analysis of cerebrospinal fluid field tests involving transgenic mice models and C. elegans models. So the topic that I chose to investigate is the efficacy of cerebrospinal fluid field tests and other related studies in detecting blood-based biomarkers, including amyloid beta plaques and neurofibrillary tangles in the brains of Alzheimer's disease patients. And so the specific research topics that I wish to target within this major general topic are: what is the effectiveness of the current tests, including CSF and lumbar punctures? Are these tests actually efficient in diagnosing Alzheimer's disease? And what are the biomarkers that CSF tests particularly target? And if these tests are proven to be less reliable and more invasive to the patient's health, what is the need to develop new tests to diagnose Alzheimer's disease more accurately? The main objective of my research project is to basically analyze the current diagnostic mechanisms that exist right now and discover if they're actually reliable or not in diagnosing Alzheimer's disease. So, overview of past research. So, Alzheimer's disease is a progressive prion neurodegenerative disease, which increases in severity with age. It is also the most common form of dementia worldwide. Early diagnosis of Alzheimer's disease can be particularly helpful for the patients, as it could potentially reduce the severity of the disease. An accurate Alzheimer's disease diagnosis can be done by identifying specific biomarkers or biological markers in certain regions of the body and brain of the patient. There are various types of biomarkers that can be used to diagnose Alzheimer's disease, including blood-based biomarkers, biomarkers in the tissues of the cerebral cortex of the patients, and also biomarkers in cerebrospinal fluid, which is the spine of the patient. So the two major types of biomarkers that are closely associated with Alzheimer's disease are neurofibrillary tangles, or tau, which is pictured here in C, and amyloid plaques, which are pictured here in F. So even though there are multiple biomarkers that could be used to diagnose Alzheimer's disease, CSF biomarkers, which are the cerebrospinal fluid biomarkers, are widely discussed as one of the most reliable in working groups or hospitals, and are also included in international guidelines for clinical practice of medicine. So these cerebrospinal fluid biomarkers are tested on several animal models, which include the zebrafish, which is pictured right here, Drosophila melanogaster, which is pictured right here, which is also known as the fruit flies, C. elegans, or roundworms, which are pictured here, and transgenic mice models, which are pictured right here. For my particular study, I decided to particularly focus on C. elegans and transgenic mice models. So the first attempt to actually create a transgenic model for Alzheimer's disease was based on the amyloid hypothesis. So in this particular study, the isoform beta amyloid precursor protein 571 was overexpressed, and it either contained the protease inhibitor domain, the human amyloid precursor protein C100 fragment, or the entire amyloid precursor protein sequence. The transgenic mice models that were used for this study developed one of three different mutations, which either exhibited hyperphosphorylated tau that can potentially develop into the neurofibrillary tangles that we see here, or APP-SWE, which is the amyloid precursor protein SWE, and PS1M146V. So these mice that were used for this study presented amyloid beta pathology at 6 months of age with increased levels of A-beta 40 and A-beta 42 and the development of amyloid plaques. So after years of studies after this particular research study, more recently, transgenic mouse tau gene has also been able to be replaced by the human tau gene in the cerebrospinal fluid samples. And these are very useful in clinical trials because it could potentially result in finding a cure for the disease in the future. So these particular cerebrospinal fluid samples in the clinical trials indicate decreased glucose levels, increased intercellular fluid pressure, high WBC or white blood cell counts and protein levels in patients that potentially have Alzheimer's disease. So I reviewed over 60 plus research articles and reviews to conduct my research project, but the two most important ones that I really want to point out are the McCann and Albert revised criteria for biomarker detection. And this particular research paper was a very big milestone in diagnosing Alzheimer's disease and other prion diseases, including mild cognitive impairment. And so this particular criteria mentioned that biomarkers are considered one of the most appropriate diagnostic methods. And this particular criteria consists of categories that are the most commonly used criteria in diagnosing MCI and Alzheimer's disease. So table 1 of the diagnostic criteria is pictured here. The second research paper that I want to point out is the C. elegans in mouse models case study that was conducted in the Karen Lab in the Keck School of Medicine, University of Southern California. This particular study used lumbar puncture, which is actually a form of extracting CSF liquid. And this particular procedure was used to determine the amount of A beta 40 and 42 levels in the CSF fluid of mouse and C. elegans models. And that actually is pictured right here. So we can see a surgical procedure of a mouse brain, and this is the syringe that was used to conduct the procedure. So they would insert the syringe into the area right above the medulla oblongata, below the cerebellum, in the relative region of the pons, and they would extract the cerebrospinal fluid to study potential Alzheimer's disease pathologies. So analyzing all the data from the various research papers, the 60 plus research papers that I studied, I concluded that cerebrospinal fluid tests are actually really reliable and can convey actually really good results. So the CSF tests of patients with Alzheimer's disease show that they have a high opening pressure, which means the intracellular fluid between the cells of their spine are flowing in a very high pressure environment. And also the CSF liquid is slightly brown with traces of blood, has high levels of protein, low levels of glucose, and a high white blood cell count. And this is pictured in this table right here. Even though CSF tests do reveal accurate results in diagnosing Alzheimer's disease, there were several challenges that were observed during the testing process. And this includes pre-analytical factors during testing, varied interpretations of the data between clinical centers, reliability, and specificity. So many of the pre-analytical factors that differ during testing include the type of sample collection, the storage tubes that were used, storage temperatures, delayed freezing of the samples, long-term stability of the organism, contamination of liquids, volume of storage samples that differed. So concluding my research paper and answering the investigative question as to the efficacy of CSF tests and other pilot studies in detecting blood-based biomarkers in the brains of Alzheimer's disease patients, I actually created two separate data charts that are pictured right here. The left data chart here, I have listed all of the molecules that can be observed in cerebrospinal fluid tests, and I compared patients with Alzheimer's disease, frontotemporal dementia, and prion diseases. And we can see right here that many molecules are actually observed in Alzheimer's disease patients, which can indicate that cerebrospinal fluid tests are really reliable and are accurate in diagnosing Alzheimer's disease. The same can be observed in this chart to the right, where I specifically picked 4 separate molecules or proteins that are found in cerebrospinal fluid tests, including proorexin, LAMP1, transliteratin, and ENPP2. And we can also see here that 80 has the highest concentration of these proteins, which can also indicate that CSF is reliable and can provide accurate results. My next steps is comparing the efficacy and reliability of CSF tests, which is this one that I research right now, with other procedures such as positron emission tomography scans and magnetic resonance imaging scans to further sharpen the diagnosis of Alzheimer's disease and compare which one is least invasive and more reliable. My research, so in the past, I've actually conducted a primer, and I've written a primer about Alzheimer's disease, and I presented it to professors at UC Berkeley. But the research experience here at Gifted Gabber was more beneficial and more thrilling. I would especially like to thank 3 separate people in this process. The first one being Dr. Jobin Varkey, whose expertise and experience really helped me to understand the content and improve my paper. He was very approachable, and whenever I had any questions, he was always accommodative, which helped me to really sail through this process in a smooth manner. I would also like to thank Professor Professor Virgil, as his knowledge and experience really helped me to improve my writing skills and organize my paper. And definitely last but not least, I would like to thank Coach Joe for giving me this opportunity to conduct another research project. And thank you everyone for your attention.
Amazing, Ria. Amazing. I can't believe the amount of work that you put in, your dedication and your never give up attitude. Going out of the four walls of your school, to take it out and follow it. It's a big round of applause. You can see the level of detail that is there, the pictures that you've drawn, the graphs and everything. It is your baby, right? And a big round of applause. Dr. Jobin, questions for Ria?
Yeah, I'm like, as I see one by one, each one of, when I see the presentation, it's very impressive. And I can't even imagine when I was in my high school to do something like this, what you guys have done. Um, Having said that, Ria, can you comment on sensitivity and specificity aspect of these biomarkers, the studies that they are doing, how sensitive they are or how specific they are? Uh,
Yeah. So when you're doing research in different lab centers, certain lab centers, even though they're using the similar animal models, such as C. elegans
mhm.
or transgenic mouse models, they can have differences in terms of their health or in terms of their physical appearance or in terms of their general bodily structures. And these can change the specificity of the data and can cause slight alterations in the data charts that are produced after the study. And in terms of reliability, this can also cause changes in that as well, because ultimately, if you don't use the same exact mouse for every single study, you're not going to get perfectly accurate results and you will get outliers in your data.
Okay, so the research is more into finding something which is very specific, so that
Mhm.
you can be sure about that it is
Yeah.
related to Alzheimer's and nothing else.
Mhm.
Okay, good, very good.
Thank you.
Excellent.